Stability testing is a cornerstone of pharmaceutical quality assurance. It involves evaluating how a drug product's quality changes over time when exposed to different environmental factors like temperature, humidity, and light. The main goal is to ensure that the medication remains safe and effective throughout its shelf life, from the moment it's manufactured until the last dose is used. This process is not just about unopened products, it also considers what happens after a product is opened and used over time. By understanding these changes, manufacturers can set appropriate storage conditions and expiration dates, ultimately protecting patient health. The primary purpose of stability testing is to assess how a pharmaceutical product's quality changes over time, especially under various environmental conditions. This ensures that the drug's shelf life and recommended storage instructions are both safe and effective for consumers. Importantly, stability testing also extends to what happens after a product is opened, which is crucial for products used over extended periods. By simulating real-world usage, stability testing helps determine how long a product can be safely used after opening, ensuring its critical quality attributes are maintained. Stability testing is especially important for multi-dose pharmaceutical products, such as liquids, creams, and ointments, which are used repeatedly over time. Once opened, these products are exposed to air, light, and repeated handling, all of which can impact their quality. Without proper testing, there's a risk that the product could degrade or become contaminated, potentially harming the user. Stability testing helps manufacturers determine how long a product remains safe and effective after opening, and whether the packaging provides adequate protection. Several regulatory agencies, including the World Health Organization, WHO, and Brazil's ANVISA, have issued guidelines emphasizing the importance of stability testing for pharmaceutical products. These guidelines ensure that products remain safe and effective throughout their intended use, even after opening. However, the guidelines are often broad, so additional recommendations are needed to address specific scenarios, such as multi-dose packaging and extended use. This is where detailed advisory documents come in, providing manufacturers with practical steps to design and conduct robust stability studies. The scope of stability testing focuses primarily on multi-dose pharmaceutical products intended for use over an extended period. This includes items like oral liquids, creams, and ointments that are not consumed immediately after opening. Single-dose products, such as blister packs or sachets, and products that must be used right after reconstitution, are generally excluded from this type of testing. By narrowing the focus, manufacturers can tailor their testing protocols to the unique challenges of products that are opened and used repeatedly. To ensure a multi-dose product remains effective and safe after opening, several principles guide the design of a stability study. These include considering the size and type of packaging, assessing the risk of microbial contamination, and understanding how the product is intended to be used. For example, larger containers may be more prone to degradation due to increased air exposure and handling. The study must also evaluate the effectiveness of preservatives and the product's ability to maintain its quality over time. In designing a stability study, the size and type of packaging play a critical role. If a product is available in multiple container sizes, the study should focus on the largest container, as it presents the greatest risk for degradation and contamination. Packaging features like resealable or hermetic closures and the use of desiccants are also evaluated to determine how well they protect the product after opening. This approach ensures that the product remains safe and effective, even in the most challenging scenarios. Multi-dose products are particularly vulnerable to microbial contamination after opening. A thorough microbial risk assessment is essential to understand how exposure to air, repeated handling, and other factors could influence microbial growth within the product. Key considerations include the effectiveness of the preservative system and the product's ability to maintain its integrity over time. This assessment helps determine the level of microbial testing required and guides the design of the overall stability study. Intended use of a pharmaceutical product, whether it's for chronic, daily use or occasional as-needed use, affects the design of the stability study. Products used daily may require more rigorous testing to ensure they remain stable throughout frequent handling and exposure. In contrast, products used less frequently might have different stability requirements. By tailoring the study to the product's usage pattern, manufacturers can ensure accurate and relevant results. Designing a stability study involves simulating how consumers will actually use the product. This means testing under conditions that reflect typical usage patterns, such as opening and closing the container, exposure to air, and handling. The study should include several critical elements, like selecting representative batches, setting appropriate storage conditions, and choosing relevant test parameters. By closely mimicking real-world use, the study provides meaningful data on product stability. Regulatory guidelines recommend testing at least 2 batches of the product, one that is freshly manufactured and one that is nearing the end of its shelf life. This approach ensures that the product performs reliably both at the beginning and end of its life cycle. If only one batch is available, samples can be split and tested at different points in time. Some authorities may require testing 3 batches for even greater reliability. Product should be stored under conditions that reflect the climate zones where it will be marketed. For example, standard conditions might be 25 degrees C with 60% relative humidity, while products intended for tropical climates may be tested at 30 degrees C with higher humidity. For particularly vulnerable products, even more extreme conditions may be used. This ensures that the product remains stable and effective no matter where it's sold. Depending on the dosage form and intended use, different parameters are tested during a stability study. Chemical testing includes assaying active ingredients and preservatives, as well as monitoring pH and related substances. Physical testing examines attributes like appearance, viscosity, and packaging functionality. Microbiological testing evaluates microbial limits, antimicrobial effectiveness, and water activity. A risk-based approach helps determine which tests are most critical for each product. Sampling process should closely mimic how consumers use the product. For example, if a product is intended for use over a month, samples are taken at the beginning and end of that period, with possible intermediate points for more detailed data. This approach helps capture any changes in quality that might occur during typical use, providing a realistic assessment of the product's stability. Once all data is collected, it must be carefully evaluated to identify any trends or significant changes in product quality. If substantial changes are detected, such as a decline in preservative effectiveness or an increase in microbial contamination, an appropriate in-use shelf life should be applied to the product label. This ensures that consumers are informed about how long the product can be safely used after opening. In some cases, stability testing may not be required. For example, if a product is consumed quickly or is chemically and microbiologically stable with excellent packaging protection, additional testing might not be necessary. However, companies should always conduct a thorough risk assessment and consult with regulatory agencies to ensure all potential risks are considered before skipping stability testing. After understanding the core principles, the next step is to design and implement a stability testing protocol. This protocol should replicate the actual conditions the product will face once opened, considering environmental factors, user habits, and repeated exposure to air and light. The protocol outlines the steps, test criteria, and examples needed to maintain compliance with regulatory expectations. Key element in protocol design is testing the worst-case scenario. This means selecting the largest available container and the longest period the product might be used after opening. Larger containers have greater air exposure and more frequent handling, increasing the risk of degradation or contamination. By focusing on the worst case, manufacturers ensure the product is robust under the most challenging conditions. Regulatory guidance typically recommends testing at least 2 batches, with a preference for 3 if available. One batch should be newly manufactured, while another should be closer to the end of its shelf life. This approach captures any variation in product quality throughout its life cycle, ensuring that the product remains safe and effective from start to finish. Next step is establishing storage and testing conditions that reflect real-world environments. For example, room temperature, 25 degrees C/60% RH, is standard, while higher temperatures and humidity are used for products sold in tropical regions. Extreme conditions, like 40 degrees C/75% RH, may be necessary for particularly vulnerable formulations. These conditions ensure the product's stability across different markets. Physical and chemical stability must be tested throughout the in-use period. Common tests include assaying active ingredients to ensure their concentration remains within specification, preservative efficacy testing to measure the ability to inhibit microbial growth, pH monitoring for chemical stability, and checks for changes in viscosity and appearance. These tests help ensure the product remains effective and acceptable to consumers. Microbial testing is critical for ensuring that the product remains safe for consumers after opening. This involves microbial limits testing to determine the total viable count of microorganisms at various points during use, and antimicrobial effectiveness testing for products containing preservatives. These tests confirm that the product does not become contaminated or unsafe over time. Implementing a stability testing protocol involves several well-defined steps. First, an initial assessment is conducted at the start of the test, with baseline measurements of all key parameters. Then, intermediate sampling is performed at set intervals, such as day 7, 14, and 30, to track any changes. Finally, a comprehensive evaluation is done at the end of the in-use period to confirm the product's stability and quality. At the beginning of the stability study, samples are opened and subjected to their first round of assessments. Key parameters like active ingredient assay, pH, viscosity, and microbial load are measured to establish a baseline. This initial data serves as a reference point for all subsequent testing, making it easier to detect any changes over time. During the study, samples are tested at various intervals to simulate typical consumer usage. For example, tests might be conducted on day 7, day 14, and day 30. These intermediate checks help identify any early signs of degradation or microbial growth, allowing manufacturers to address potential issues before they become significant. At the end of the testing period, a final evaluation is conducted to ensure that no significant deterioration has occurred. All key attributes are tested again, including active ingredient levels, preservative effectiveness, and physical properties. This comprehensive assessment confirms whether the product remains safe and effective for consumers throughout its intended in-use period. Once all testing is complete, the data must be carefully interpreted to identify any patterns or significant changes. For example, a loss of potency, breakdown of preservatives, or changes in appearance could indicate that the product is no longer suitable for use. Based on these findings, manufacturers can make informed decisions about labeling and recommended usage periods. If significant changes are detected during stability testing, manufacturers may need to update product labeling. For example, a product might require a warning to discard after 30 days once opened, or it may be deemed safe for use throughout its entire shelf life. Accurate labeling ensures that consumers are properly informed and protected. Topical creams and lotions. A typical stability testing protocol might include chemical assays every 7 days for 30 days, microbial testing at day 0, 15, and 30, and regular checks for changes in color, odor, and viscosity. These tests help ensure that the product remains safe, effective, and pleasant to use throughout its intended period. For liquid oral suspensions, the protocol might involve assaying active ingredients at day 0, 14, and 30, microbial testing every 14 days, and physical testing for sedimentation or separation at each interval. This comprehensive approach ensures that the suspension remains stable and safe for consumption throughout its use. Thorough stability testing is essential for ensuring the continued safety, efficacy, and quality of multi-dose pharmaceutical products after opening. By following a structured and comprehensive protocol, manufacturers can confidently label their products with accurate usage instructions, ultimately safeguarding public health and consumer trust. Robust stability testing plan not only protects consumers, but also helps manufacturers meet regulatory requirements. By adhering to guidelines from agencies like WHO and AMVSA, companies can ensure their products are accepted in global markets and avoid costly recalls or compliance issues. Stability testing is not a one-time process. As new formulations, packaging, or usage patterns emerge, protocols should be updated to reflect the latest science and regulatory expectations. Continuous improvement ensures that pharmaceutical products remain safe and effective for all users. At the heart of stability testing is a commitment to consumer safety. By rigorously evaluating how products perform after opening, manufacturers can prevent adverse events and ensure that every dose delivers the intended benefit. This focus on safety builds trust and supports public health. Summarize effective stability testing involves selecting appropriate products and batches, simulating real-world storage and usage conditions, conducting comprehensive chemical, physical, and microbial tests, interpreting data carefully, and updating labeling as needed. Each step is crucial for ensuring that pharmaceutical products remain safe and effective throughout their lifecycle. Leading pharmaceutical companies follow industry best practices by collaborating with regulatory agencies, adopting risk-based approaches, and continuously updating their testing protocols. These practices help ensure that products meet the highest standards of quality and safety, no matter where they are sold. Packaging plays a vital role in maintaining product stability. Features like hermetic seals, resealable closures, and desiccants can significantly extend the safe use period of a product after opening. By investing in high-quality packaging, manufacturers add an extra layer of protection for consumers. Thorough risk assessment is essential before deciding whether stability testing is necessary. This involves evaluating the product's formulation, packaging, intended use, and potential for contamination or degradation. If risks are low and the product is well protected, testing requirements may be reduced, but documentation and regulatory consultation are still important. Stability testing protocols are shaped by global guidelines from organizations like WHO, AMVSA, and the FDA. These guidelines provide a framework for ensuring product safety and efficacy, but often require adaptation to specific products and markets. Staying informed about regulatory updates is key for compliance. Educating consumers about proper storage and usage of pharmaceutical products is an important part of stability testing outcomes. Clear instructions on labels, such as storage temperature and discard dates, help ensure that products are used safely and effectively, reducing the risk of adverse events. Even after a product is launched, continuous monitoring of stability data is important. Post-market surveillance helps identify any unexpected issues and allows manufacturers to update protocols or labeling as needed. This ongoing vigilance is essential for maintaining consumer trust and regulatory compliance. As technology advances, new methods for stability testing are emerging, such as rapid analytical techniques and digital monitoring. These innovations promise to make testing more efficient and accurate, helping manufacturers respond quickly to changes and maintain high standards of quality. Collaboration between manufacturers, regulators, and industry associations is key to advancing stability testing practices. Sharing data and best practices helps the entire industry improve, leading to safer and more effective pharmaceutical products for everyone. Stability testing is a critical process that ensures pharmaceutical products remain safe, effective, and high quality throughout their intended use. By following robust protocols, staying informed about regulatory changes, and prioritizing consumer safety, manufacturers can deliver products that truly benefit public health. In conclusion, stability testing is not just a regulatory requirement, it's a vital part of pharmaceutical development and consumer protection. By investing in comprehensive testing and continuous improvement, the industry can ensure that every product delivers on its promise of safety and efficacy, from the first dose to the last.